Okay, it's 9 o'clock, time for the seminar. I am very happy to introduce our next lecturer from industry, Ludovic Bourre. I believe that this lecture would be helpful for project proposals which you could submit then to any funding and principally you could get additional knowledge how to work with in vivo models and how the Crohn Bio could help you in your projects in future. Ludovic, you're welcome.
Yeah, thanks, Ira, for the introduction and thanks to have the opportunity to present this topic this morning. So today I'm going to talk about advancing immuno-oncology therapy and what are the next generation syngeneic models. So during this presentation, I will address a few topics on syngeneic models, starting first with immunoprofiling, then looking at immune checkpoint inhibitors treatment and dissecting the mechanism of action, how we can use a syngeneic model to address those questions. Then I will talk about engineer a mouse model as a tool for mechanism of action study. And I will conclude talking about engineer syngeneic lines and also what we call UGEM, which are KI models that are humanized for immune checkpoint targets. So today the main focus in R&D and particularly in clinical try to focus on those areas that are listed here. So basically enhance T-cell activity, increase the immunogenicity of the tumor, release immune brakes mechanisms, change immune cell polarization, and promote T-cell trafficking into the tumor. But the key word here, I would say, is a combination. Everyone now is looking at combination strategy, are using different treatment modalities, both at preclinical and clinical stage. And actually, in clinical, if we, based on this review that have been published last year, you can see that there is about 3,000 clinical trials built on the pillar of PD-1, PD-L1 blockade. And as you can see, there is a dramatic increase between 2017 and 2019 with a large number of increase of clinical trial looking at those combination strategy. So that's mean that there is a lot of resources that are used in a clinical trial, trying to figure out a more efficient way to treat patients. So the question is, can't we better use preclinical model to address some of those questions? So there is different mouse model that can be used to investigate immunotherapies. The first one are here at the GEM mouse model that have been developed to provide a better model of human cancer. They are specially designed around genetic events observed in human disease with transgenic knock-in or knockout model based around oncogene or tumor suppressor. So there is a wide range of well-characterized models that are available now with a clear molecular pathogenesis of disease. So the advantage of those models basically is have natural tumor progression, also a natural tumor microenvironment. As I mentioned, they have relevant genetic driver, and they have also intact immune system because they are fully immunocompetent, but it's still a mouse immune system. The challenge around this model is that the tumor is non-synchronized, so it's very difficult to use those mice in a large cohort for efficacy study. Also, they are logically challenging. License usually those models are under license, so that could be expensive, and also there are few neoantigens, so those tumors are quite cold tumor and basically very resistant to immunotherapies. A second approach that can be used to test immunotherapies is the humanized mouse model. In this case, you can test human antibody, and you can also grow human tumor, either patient-derived xenograft or conventional xenograft. However, those humanized mice have suboptimal immunity. Not all immune cells are represented in those mice. Also, most often there is allogenic immune response. The mice are quite expensive, and those mice also subject to graft versus of disease, and also graft versus tumor. And finally, I would say this is the workhorse in the preclinical space, syngeneic model. Those models have been largely published in the literature. They have intact immune system, and they are easily accessible. They are great for screening, and also, as I mentioned, this is kind of the industry standard for preclinical work. However, there is also some, I would say, challenge. They are partially predictive. They are mostly carcinogen driven, so it's not, I would say, natural tumor progression. You can test only mouse surrogate, and there is no chronic inflamed tumor environment in those models. So now I'm going to show you some work that we have conducted looking at in-depth immunoprofiling of the syngeneic model in order to understand better what are the different, I would say, differences between the different syngeneic models that can be used. So here, this is immunoprofiling that we have conducted on tumor-bearing animals when at baseline, so basically when the tumor volume was 100 millimeter cubes, and we have tested here a panel of syngeneic models. So maybe it's a little bit small here, but as you can see here, the immune cell infiltration could be very different. For example, in EMT6 model, you have a high amount of CD45, whereas some model like B16F10, they are very low infiltration. And also you can look after a different immune cell compartment and look at model that may be of interest for you. For example, a model that show high Treg component or high macrophages. So basically, when you choose a model, you can choose the model based on those characteristics according to what type of question you want to answer. So in the previous slide, what I show you is at the baseline, but actually the immune cell infiltration within the tumor is a very dynamic process. So if you look at the baseline, or if you look later, you can see that the immunoprofile change during the tumor course. So in this case, I'm going to show you two head-to-head comparison that we have done on two different tumors. So one in MC38, which is a colorectal cancer, and one EPA16, which is hepatocarcinoma. And we have looked at a different time points, so at baseline, and also 7 days, 14 days, and 28 days after tumor engraftment. And we have treated those tumor either with, so the PBS, this is a control group with the isotype control, and also with PD-1. So you can see here for MC38 with anti-PD-1 treatment, we have like intermediate efficacy around 60% of tumor growth inhibition, whereas in EPA16, we do see very strong response here with tumor regression compared to the control group of the isotype control. So if we look here on the EPA16, so on this graph, this is the tumor volume according to time. So each dot represents one tumor. So you can see clearly here that the anti-PD-1 treatment is very efficacious. After 31 days, the tumor is very well controlled, whereas in the PBS group, the tumor is growing nicely. And so if we look at the dynamic change, you can see that EPA1 tumorigenesis is associated actually with increase of CD8 T cells, but also increase of NK cells during the course of the study, and also a shift from M1. So there is a decrease in M1 in the PBS group and increase in M2. And this profile is consistent with a strong cytolytic T cell immune response observed in this model. So actually, when you treat with anti-PD-1, you can see that you increase significantly the CD8 infiltration, and also that you decrease the M2 macrophages in this model under PD-1. Now, if we look at the MC38, so same here, this is the tumor where there is definitely less efficacy of anti-PD-1 in this model. Now, if we look at the PBS group, you can see that the dynamic here of the change is not as obvious compared to the EPA1/6. So basically, the model doesn't really change in terms of immune profiling across different time points. However, when we treat with anti-PD-1, we do see some delay increase in CD4 and CD8 after 13 days, and also some decrease in the macrophages at a later time point. So in addition to these two models, actually, we have done this exactly the same profiling on 13 syngeneic cell lines, which are listed here. So we have done the same immunoprofiling to understand better what is the dynamic of those models in terms of immune cell changes according to the tumor growth. So in addition to the immunoprofiling data, we have also benchmarked the syngeneic model with immune checkpoint treatment and look if there is different mechanism of action between different models with the same antibody immune checkpoint inhibitor. So I'm going to show you one example with anti-PD-1 treatment. So here, this is basically the tumor growth inhibition that are plotted here on those waterfall plot. So here we have tested anti-PD-1, anti-PD-1, and anti-CTLA-4 across a range of the syngeneic model that are listed here. So the blue bar are the one that have been tested several times, and the one in orange, that means we have just done tested this model once with this immune checkpoint. But I think what is important here, so you can definitely see that syngeneic model have very different response with the immune checkpoint inhibitors, and they behave very differently between the different model, either for anti-PD-1, PD-1, and CTLA-4. So in order to, as I said, to understand better how immune checkpoint inhibitors work and how those models can be used to understand better the mechanism of action, and also work on combination strategy, here we have performed some study and done some immune cell depletion and see what is the influence of different immune cell compartment on the efficacy of the anti-PD-1 treatment. So in this example, we have tested 4 models, so MC38, EPA1/6, another colon model, CT26, and EMT6, which is a breast cancer model. And so we have treated this model with PBS or with anti-PD-1. So the PBS is the blue curve, and the anti-PD-1 is a yellow curve. And we have also so treated the same anti-PD-1 treatment, but we have performed some depletion of certain immune cells. So we have deplete CD8, CD4, NK cell, macrophages, and Treg. And as you can see here, the immune cell depletion could have a very strong effect on the efficacy of the treatment, and very different effect actually if we compare the different models. So for instance, if we look at the MC38, when we deplete the CD8, we completely abolish the anti-PD-1 treatment. So you can see the gray here, which is PD-1 plus CD8 depletion, the same level compared to the PBS group. However, if we deplete CD4 macrophages or CD25, which is the Treg, we do see improvement of the anti-PD-1 treatment. Now, if we compare with EPA16, actually, this is quite of the opposite effect. If we deplete CD4 here, we completely also abolish the anti-PD-1 treatment, even though the tumor is growing faster compared to the PBS group. And the other, I would say, immune cell depletion has no so much effect here on the anti-PD-1 treatment. So this is so those results basically are summarized here. So we have looked here at the tumor growth inhibition impact with immune cell depletion compared to the anti-PD-1 treatment. So what is represented here is tumor growth inhibitor percentage change compared to the anti-PD-1 treatment. So that's mean when the bar here is going below that, the tumor is growing faster compared to the anti-PD-1 treatment. And when this is higher here, that means that the treatment, anti-PD-1 treatment is improved compared to anti-PD-1 alone. So we can clearly see here for MC38, when we deplete CD8, we completely abolish anti-PD-1 treatment, whereas when we deplete CD4 macrophages or Treg, we increase the efficacy of anti-PD-1, which is also, I would say, the same profile for CD26 and AMT6. We do see very similar trends with a strong dependence of CD8 regarding the efficacy of anti-PD-1 treatment. However, we can clearly observe here that for EPA16, this is a completely different mechanisms. Basically, there is no improvement or whatsoever depletion we use, and with CD4 depletion, this really impact the PD-1 treatment. So in order to understand what I would say immune cell population are modulated under depletion, so we have PD-1 treatment group. So the red number, for example, here indicate statistical significant change, and the red box, this is an increase, and the blue box a decrease. So if we look here, so on the left, this is a different immune cells population. And for each model, so the first column here, this is anti-PD-1 plus CD4 depletion. The second one, it's anti-PD-1 plus CD8 depletion. The third, this is macrophage depletion. And the third, this is NK cell depletion. So if we look at MC38, for example, if we deplete CD8, so we can see here that we have a very strong CD8 depletion, because we are almost at 100% reduction of the CD8 population, so very good depletion of the CD8. We can clearly see that there is a strong increase of macrophages, which is also the case for the model that show very similar behavior compared to MC38 for MT6 or CT26. We do see very strong increase on macrophages. Also for MC38, when we deplete CD4, we can see here a very strong increase of CD8 infiltration. Now, if we look at the HEPA-1/6, we can see here that CD8 depletion, there is not much influence in terms of immunomodulation. However, when we deplete CD4, we can clearly see that we increase the macrophages and also the MDSC population. So I won't go through all the details here. This presentation will be shared afterwards, but there is a lot of information that you can look at in terms of immunomodulation. But I would say the one I have described is the main outcome of those observations. So after this experiment, actually, we have look at deep proteomic analysis to also try to understand what modulation is involved in this different behavior between the different models. So for this, we have compared HEPA-1/6 and MC38, and we have done some comparison between the group 1, 2, and 3, which is PBS and TPD1. Anti-PD1 under CD8 depletion and anti-PD1 under CD4 depletion. So from this unsupervised clustering here, you can see that the two models, so the HEPA-1/6 in red and the MC38 in blue, they are clustering very differently between the two strains here. What is kind of striking here that in group 4, so with anti-PD1 and CD4 depletion, for MC38, the model also cluster here. And this is here the protein level, so with increase of the protein when this is in red and decrease of the protein when this is in blue. So we have looked at different actually pathway and different readouts. So the first thing that we have looked at the effect of CD4 depletion on antigen presentation on cell toxicity. So we have looked at different genes in antigen presentation on MHC class 1, also MHC class 1 expression on cytotoxicity. So for MC, so the first 4 boxes here as for HEPA-1/6, so the PBS group, the PD1 plus CD8 depletion and PD1 plus CD4 depletion. And the 4 last boxes, this is exactly the same group, but for MC38. So what we can see here that CD4 depletion in MC38 upregulate MHC class 1 protein for years for the different protein, where this is actually the opposite effect for HEPA-1/6. When we deplete CD4, we decrease antigen presentation. Same here observation for MHC class 1 expression. When we deplete CD4 in MC38, we increase MHC class 1 expression. And we also increase the cytotoxicity in MC38, whereas is decreased in HEPA-1/6. Also, we have a look at immunomodulating signal. So looking at, for example, inhibitory signal like PDL1, PDL2, LAG3, VSTA, also IDO1, CD39, those two, and also some activation signal. So what we can observe here is it's also that HEPA-1/6 have a higher baseline expression of immunosuppressive markers. So we can clearly see that HEPA-1/6 have higher expression of PDL1 compared to MC38, and this is true for all the markers here. And now when we deplete CD4, we decrease those suppression marker in HEPA-1/6, which is a little bit here counterintuitive. However, this increase in MC38. Regarding the immunoactivation signal, there is not really some change observed in those pathways. And last but not least, actually, we have looked at so the impact of the CD4 depletion, and what we have found is that this is boosting the interferon gamma signaling pathway. So we have looked at a range of proteins that are involved in interferon gamma pathway. So they are labeled here in red, and what we can clearly see in MC38, when you deplete CD4, you increase significantly the interferon gamma pathway. That could explain why you have higher efficacy of anti-PD-1 treatment after CD4 depletion in MC38, whereas this is the opposite effect in EPA16, where we don't see, where actually we do see some decrease on the interferon gamma for EPA16 when we deplete CD4. So in summary, what we can conclude here from those observations is that those two models have very different behavior and respond very differently under anti-PD-1 treatment. With MC38, so a very strong dependence on CD8 to respond to PD-1, and that CD4 depletion in this model lead to enhanced response of anti-PD-1 treatment in this model. Whereas EPA16 response to anti-PD-1 is not dependent on CD8. However, CD4 depletion leads to a resistance of the tumor. We have seen when we deplete CD4, we increase immunosuppressive cells like MDSC or macrophages. So there is still some question open. Is this tumor difference is due to lower immunogenicity or impaired interferon gamma signaling? Also, is it due to immunosuppressive tumor environment change? So those questions are still open, but I think the conclusion here is quite clear and striking that those two models have very different behavior. So now I'm going to talk about a few models that we have developed also to understand the mechanism for action. So they are based on genetic engineering mouse model. So those models here that have been developed in order to, I would say, in addition to the antibody depletion that I've just show you, you can use diphtheria toxin receptors, so the DTR germline, to do the same type of work. So we have developed DTR mouse model again for different immune cell compartments. So with CD19, CD8, NK cells, FOXP3, and CD4 DTR line. So basically, when you treat those mice with this diphtheria toxin that induce toxicity in the dedicated immune cells that are engineered to express the DTR receptor, and you deplete completely the immune cell compartment. So in this example here on the CD19, you can see that after diphtheria toxin, so this is CD19 in the blood, you can see a strong drop here, almost zero CD19 in the blood after the diphtheria toxin. And here, this is the control group. And you can look also at the impact of the immune cell depletion of the tumor growth. So you can see here for CD19, there's not much impact of depletion on the tumor growth. So this is MC38 here. However, if we take as an example here, CD8, when we deplete CD8 population, we can see here that the tumor is growing much faster. So it's less control compared to the control group. Same here, when you deplete FOXP3, actually, you get a better control of MC38 with spontaneous tumor regression when you deplete the Treg in MC38 compared to the control group. So as I mentioned, you can use also antibody, and this has been shown in the previous slide. But you can see some differences actually between antibody depletion or the DTR approach, particularly for our FOXP3. If you I used CD25 here to deplete the Treg in EPA16, you can see there is no much different here when we deplete anti-CD25 with anti-CD25 when we deplete and combine with anti-PD-1 treatment, there is no difference with anti-PD-1 treatment alone. And also there is no difference in the control group, the PBS or the PBS plus with depletion of Treg. So basically there is no much impact here on the depletion on either the growth of the tumor or the efficacy of the PD-1 treatment. However, if you deplete with the FOXP3, with the DTR line, you can see very different actually outcome. If you just treat the mice with diphtheria toxin, you can see that the tumor is very well controlled after FOXP3 depletion. And also, when you combine anti-PD-1 treatment plus FOXP3 depletion, you increase the efficacy of the anti-PD-1 with even more efficacy observed after depletion of FOXP3 under anti-PD-1 compared to anti-PD-1 alone. So very different outcome between the two different approaches. And that could be explained because anti-CD25, in addition to target Tregs, also eat effector T cells on the CD4. And in this case, that failed to show tumor suppression. However, in the Treg DTR line, that led to significant tumor growth inhibition and also enhanced anti-PD-1 efficacy. In addition to those DTR line, we have also developed our immune cell reporter line. So those lines are engineered to you can use those lines to look at specific immune cell populations. So for example, we have developed CD8 that express luciferase on EGFP. So you can basically monitor the CD8 infiltration in the tumor. So for example, if we engraft the CD8 luciferase mouse with MC38 tumor, so this is no treatment. So you can see here some immune cell infiltration, CD8 infiltration in the tumor. However, if you treat with anti-PD-1 treatment, you can see an increase of the CD8 infiltration after anti-PD-1 treatment. We have also developed a FOXP3, so you can actually monitor also FOXP3 modulation under treatment. So those two lines are actually available, but we are working other reporting line like for NK cells, DC, B cells, and macrophages. Also, we have also developed reporter line for cytokines. So we have the reporter line for interferon gamma on HEL2. So this is here another example. So this is the white-egg mice or the interferon gamma mice with no stimulation. When we stimulate with concanavalin A for 4 hours, we can see interferon gamma release in the mice after treatment. So also we have developed engineered cell line to look at specific, I would say, antigen response. And also I'm going to talk about humanized knock-in mice that are being humanized for certain targets. So here two different examples. We have developed antigen expressing line for ovabuline or SIY. And basically those models can be used to detect ova-specific T cells response or GP70 specific T cell response by tetramers. And you can use also this model for adoptive T cell transfer experiment. So in this example, we have treated different cell line that express those antigen with anti-PD-1. So you can see that we do see very strong efficacy of anti-PD-1 in those lines that usually are more immunogenic compared to the, I would say, wild type syngeneic lines. So what we have done here is to compare MC38 compared to MC38 over and look at the immunoprofiling. So this is the tumor volume here that has been used for the facts. So you can see that the tumor volume here is very quite similar between MC38 and MC38 over. So this is a CD35 in the TILs. So you can see here in the ovalbumin, the tumor have higher increase of immune cell infiltration compared to the MC38. So that's why I've stated before is that basically those models are more immunogenic. And if we look at the different immune cell compartment, you can see that you have higher CD4 or CD8 population in those in the HOVA compared to the MC38 wild type in the TILs particularly. It's not so obvious in the blood. Now if we take the cure mice, so basically I'm going back here. So in this experiment, so we have took the mice that have been completely cured here. And we have so we have taken the blood of those mice. So what you can see here that there is antigen specific T cells for ovalbumin, and we have used tetramers here. So you have an increase in the ovalbumin specific T cell when you treat with anti-PD-1 compared to the one that has not been treated with anti-PD-1. And this is true in the lymph node and also in the spleen. And actually, when you take the blood of those mice and then you do adoptive transfer on new mice that have been engrafted with MC38, you can see that you have a very good control of the tumor growth compared to the non-treated mice. You can see here there is completely rejection of the tumor when you infuse the T cells from the mice that have been cured previously. You have also good control actually from the mouse that have been previously that was not treated with PD-1, but just only have the tumor burning. So that's mean also without any PD-1 treatment, they will be able to have a specific T cells against this tumor. And here this is just OT1 control, and basically OT1 mice are the mice that are engineered for ovalbumin, so they have specific T cells for ovalbumin. And we have looked also if we can see the same effect in MC38. So basically the cell line here does not express ovalbumin, and you can see here that you have also good control even though there is no ovalbumin expression from the anti-PD-1 or the MC38 ovalbumin mouse model. So that means you have also antigen spreading in the T cells that can control the tumor growth. So the previous example I show you basically is to understand how immune checkpoint inhibitors can be those models, how they can be used to test immune checkpoint inhibitors, but that means these systems are entirely mouse dependent. So you can't basically evaluate antibody that targets immune receptor, for example. So you need to use antibody specific models for mouse. So in this case, we have engineered U cells that express human tumor associated antigen. So in this case, you can test a human antibody that does not cross-react with the mouse targets. So we have developed syngeneic line actually that express different human cell targets. So we have the list here. So human CD20, 29, or 2 EpCAM, but we are developing much more. Actually, there are other models in development for to express those human targets, which are very relevant for bispecific, for example, antibody testing. And actually, so you can combine those U cells, so syngeneic line that express human antigen, in addition to the knock-in mice for human targets. So here there is 2 examples. So we have tested MC38 that express CD47 in human CD47 CF alpha double knock-in mice. So that means you have a completely humanized system, in this case from the mouse side, but also from the tumor side. And we have tested different here anti-CD47, so the HU5F9, and also 2 other, the 121 and the 121A. So 121A is the same that the 121, but has a less affinity for the red blood cells. So there is less adverse events with this antibody because anti-47 knows this target, it's very well known to induce some adverse events and some anemia after treatment. And we have tested the combination with anti-PD-1. So in blue, this is the vehicle group. In black, and this is the anti-PD-1 treatment. So you can see here that U5F9 shows some intermediate tumor growth inhibition. Also the 121. The 121A, it's here, and you can see there is a little less efficacy compared to the 121. However, when you combine with anti-PD-1 treatment, you do see some synergetic effect here in the combination. Another example here is MC38 with human PD-L1 that have been engrafted in CD137 knock-in mice. So in this case, same, you can test a human antibody in those mice. So we have tested urelumab for 1BB and atezolizumab for anti-PD-1, and we have looked at the combination. So you can see that the single agent, either urelumab or atezolizumab, there is no much efficacy here. However, when you do the combination, you have increase of the efficacy with quite significant tumor growth inhibition. Also, we have tested the bispecific here for CD137 and PD-L1, and we do see very strong control of the tumor growth. So in addition to those KI mice, we have also developed some transgenic mice for CD3. So this model is very useful to test bispecific CD3 engager and against also tumor-associated antigen. So in this case, we have tested this model in A20 tumor model with 2 different bispecifics. So one mouse bispecific, so CD20, mouse CD3, mouse CD20, and in the other hands, mouse CD20 and human CD3. So you can see here that both bispecific show strong B cell depletion after the first dose, and also a good control of the A20 tumor after injection of the bispecific compared to the PBS group. Here, this is another study that we have done for clients. So in this case, he has a bispecific that targets a human tumor-associated antigen, and we have expressed the same antigen on CD26, and we have tested 2 different doses of this bispecific, and you can see that there is very great dose response here with intermediate response with 0.3 mg per kilo, and very strong response with 1 milligrams per kilo. So in addition to the efficacy study that I just show you, those UGEN mice also can help to predict, I would say, adverse events that have been observed, which most of the immune checkpoint or immunotherapy in clinics. So these have been reported numerous adverse event or toxicity issue with immune checkpoint therapy or even also cell therapy. That could be, in some case, very strong and very dangerous for the patient. So one adverse event which is well known in CD137, for example, is a liver toxicity. So when patients are treated with this antibody, this induces very severe liver toxicity. So actually, we have looked at how those humanized KAHI model can address those questions at preclinical stage to look at, I would say, adverse event that could be observed before going to clinical trial. So in this example, we have tested the CD26 in the CD137 humanized mice, and we have tested different antibodies. So urelumab, which is the anti-CD137 that is used in clinic, which is IgG4. And we have also engineered produce urelumab, but on mouse IgG1 backbones to be able to have the ADCC effect on FC engagement. And also we have the DANA version, which have been so in this model, in this antibody, there is some mutation that have been performed on the IgG1 and to reduce the FC engagement. And as you can see here, there is strong efficacy of all the antibody with urelumab in yellow. Here are the two different doses. There is no much difference in terms of doses. And urelumab in blue on the IgG1 backbone, we can see that it's higher. There is higher efficacy compared to the urelumab on the IgG4 backbone. And when you have the DANA version, so with the mutation that impairs the FC engagement, you do see some here a little bit reduced toxicity, but still some effect. And here, this is just a control, I would say, group on the white-type mice with CD26 with the mouse CD137, where we see also good efficacy. And then we have looked at the liver toxicity, looking at liver enzyme and see if there is some increase in those enzymes after treatment. So what we can clearly see here is that urelumab on IgG4, which are not cross-linking with FC receptor, there is no toxicity observed with this antibody. However, when you use urelumab on the mouse version that can cross-link with FC receptor, we do see some increase of some toxicity here, which is abolished when we use the antibody version that is not engaging with FC receptor. And this is here the mouse version where we do see some liver toxicity with the mouse antibody. So definitely here we can clearly see that this model can be used to address the toxicity, but we need to be careful about cross-linking with FC receptor. In this example, we have regarding the antibody. Another second example here, which is well known, when you combine PD-1 and CTLA-4, that has been lead to severe adverse events with different, I would say, clinical outcomes, like acute lymphocytic myocarditis, joint swelling, skin inflammation, and also cytokine storm in some patient. So in this example, we have used a double knock-in model for PD-1 and CTLA-4. So those mice are humanized to express both human targets. And we have done some repeat injection of the combination in those mice. And we have done this in different mouse backgrounds, so the black cyst background and the biopsy background. So as you can see in the black cyst background, we don't see much, we don't see any adverse events, even in terms of survival or clinical observation. There is no such events observed. However, in the biopsy background, we do see some decrease in survival when you treat with the combination compared to the PBA group of the single agent. And we do see also some clinical signs with joint swelling, skin inflammation, and increase of T cells in the myocardium. As a summary here, to conclude this presentation, so syngeneic model are today the workhorse for immuno-oncology drug discovery. So as I show you, tumor microenvironment are very different between the different syngeneic model, as well as the crosstalk to the host immune system differ very significantly. And IO treatment lead to distinct response in different syngeneic model mediated by different immune cell lineage. And creation also of tools like GEMMOS model can be used for mechanism of action study. And also the UGEM model, so the KE humanized mice can be used to test human antibody agent and also address also immunosafety questions before going to clinics. So I think this is my last slide. So yeah, I would like to thank you for your attention, and I'm very pleased to answer any questions.
Okay, thank you very much, Ludovic. Edith would like to ask you, Edith.
Yes, thank you very much for this interesting talk. I wanted to ask you regarding the first part of your talk where you tested actually at the beginning of 4 different cell lines, and you saw a very large heterogeneity between the cell lines in terms of the immune contexture in the different models. And I wanted to ask you if you think that the difference is because of the differences in tumor types, in the strain of the mice, or if you will see even in the same tumor type, which is administered to one specific syngeneic model or mouse, you will see such differences. So the question is whether it is heterogeneity even between different cell types that belong to the same tumor type in the same mouse strain. As I'm asking this because I think it's relevant to the human setting, because in the human you have so many different parameters that can affect the difference between the contexture of the immune system in the tumors.
Yeah, so indeed we do see very strong, very large heterogeneity in those mice, even within one model. So if we take MC38, for example, we do see some heterogeneity between mice and also between different study. And actually we have different site across. So we have site in US, in Europe, and in China. And we do see also different I would say, behavior in terms of immune checkpoint response. And that is linked actually to the mouse vendor, because we are sourcing mouse from different vendor in those places. And also the microbiome environment have a very strong influence as well. So I would say it's, yeah, as you said, this is very true. I think so as well, that is very linked to, I would say, what we do see in the patient population. There are very different, I would say, immune profiling and immune cell profiling between patients, and we do see also this in syngeneic, even though it's less heterogeneous because we are using always the same cell line and the same mouse strain, but even though we do see some variation in terms of immune checkpoint response. For example, from MC38, I would say the mean efficacy of anti-PD-1 is around 50% tumor growth inhibition, but we can see some experiment when we're gonna have like 80% of tumor growth inhibition, and another study that would be 20. So yeah, so that is something to consider. That's why also you need to really to think about the power and the group size that need to be used in those kind of experiments to capture this heterogeneity.
Okay, thank you very much.
Does anybody have any further questions? We have the comment, create talk. What kind of models do you use in understanding the residual cells in myeloid region?
I guess you can use whatever model. I think in syngeneic model you can look at any of residual cells by flow. For instance, you can look at different immune cell population, and if you have interest in the myeloid compartment, you can definitely look at this. You can also have models to deplete specifically myeloid compartment, either DC or macrophages. As I show you, you can deplete macrophages with specific antibodies, or we are developing DTR line to deplete myeloid compartment. So you can investigate what is the influence of this specific immune cell compartment on the tumor biology, or the impact of this cell population on the treatment modalities. I don't know if I answered the question, but.
Okay, we have an additional comment from Katarina. Very interesting, and thanks a lot. And next question from Krishna Das. Did you look at the tumor mutation burden of MC38 and HIPAA tumor cell line?
Yeah, so we do have for all home model, actually, it's we have RNA-seq data, and they are fully sequenced. So we have looked actually at the tumor burden and mutation load. So we have those data in hands, and we do see some correlation with the immune checkpoint response. Basically, so the model are quite cool. Have a low tumor mutation load, whereas the other, like HIPAA-1/6, have high mutation load. Yeah.
And there is an additional question from Krishna. And did you do any kind of lymphodepletion before adaptive transfer?
No, in the study that I've shown you, we have not done lymphodepletion. So we have just infused the PBMC from the mice that I've shown very strong response, and then we just infuse in new mice. Yeah, we have not conducted any lymphodepletion in this case.
Okay, any further questions? I don't see any questions in chat. Probably anybody would like to ask using their micros. Ludovic, I don't see any further questions. Thank you very much again. And I hope that our seminar participants will contact your company, and probably they will directly contact you in order to create any study, or they would ask you about the help for planning of the research proposals or for the educational purposes or any other reasons to discuss their projects. And therefore, thank you very much again for your excellent talk. And I hope that you will attend our seminars in future as well. And all the participants are invited to the next seminar, which will be organized in February, on 25th of February. And this seminar will be devoted to retroviruses, and the lecture will be given by Claudia Matteucci. Thank you very much to all.
Thank you very much.
Thanks a lot.
Thank you.
Thank you so much. Have a great day.
Bye-bye.
Yeah. Thank you very much, and see you in 2 weeks.